Epidermal growth factor or EGF is a growth factor that stimulates cell growth, proliferation, and differentiation by binding to its receptor EGFR. Human EGF is a 60-45 DAR protein with 53 amino acid residues and 3 intramolecular disulfide bonds. History: the discovery of EGF won Stanley Cohen and Rita Levi-Montalcini the Nobel Prize in Physiology or Medicine in 1986. Function: EGF results in cellular proliferation, differentiation, and survival. EGF is a low molecular weight polypeptide first purified from the mouse submandibular gland, but since then found in many human tissues including submandibular gland, parotid gland. Salivary EGF, which seems also regulated by dietary inorganic iodine, also plays an important physiological role in the maintenance of oroesophageal and gastric tissue integrity. The biological effects of salivary EGF include healing of oral and gastroesophageal ulcers, inhibition of gastric acid secretion, stimulation of DNA synthesis as well as mucosal protection from intraluminal injurious factors such as gastric acid, bile acids, pepsin, and trypsin and to physical, chemical and bacterial agents. Biological sources: epidermal growth factor can be found in human platelets, macrophages, urine, saliva, milk, and plasma. Mechanism: EGF acts by binding with high affinity to epidermal growth factor receptor on the cell surface and stimulating the intrinsic protein tyrosine kinase activity of the receptor. The tyrosine kinase activity, in turn, initiates a signal transduction cascade that results in a variety of biochemical changes within the cell, a rise in intracellular calcium levels, increased glycolysis and protein synthesis, and increases in the expression of certain genes including the gene for EGFR, that ultimately lead to DNA synthesis and cell proliferation. Egf Family: EGF is the founding member of the Egf family of proteins. Members of this protein family have highly similar structural and functional characteristics. Besides EGF itself, other family members include heparin-binding EGF-like growth factor, transforming growth factor I+or-, amphiregulin, epiregulin, epigen, batracellulin, neoregulin-1, neoregulin-2, neoregulin-3, neoregulin-4. All family members contain one or more repeats of the conserved amino acid sequence, CX7 CX4-5 CX10-13 CXCX8 GXC, where X represents any amino acid. This sequence contains 6 cysteine residues that form 3 intramolecular disulfide bonds. Disulfide bond formation generates 3 structural loops that are essential for high-affinity binding between members of the EGF family and their cell surface receptors. Egf therapy: increased activity of the receptor for EGF has been observed in certain types of cancer, often correlated with mutations in the receptor and abnormal functions such as constitutive receptor signaling independent of the levels of EGF or of binding of EGF. Pharmaceutical drugs developed for inhibiting the EGF receptor include gefitinib, erlotinib, and afatinib for lung cancer, and cetuximab for colon cancer. Monoclonal antibodies are potential substances for this purpose. Interactions: epidermal growth factor has been shown to interact with epidermal growth factor receptor. References. Further reading. External links: Sheungsi Tsungbang Pharma Tech Company, limited supply of epidermal growth factor, EGF at the Human Protein Reference Database. Epidermal growth factor at the U.S. National Library of Medicine. Medical subject headings: EGF model in Biomodels Database.